Alright, so picking up with our third installment of our discussion on cell respiration and fermentation. Alright, so we've talked about aerobic cellular respiration. Now we're going to discuss what happens in the absence of oxygen. Alright, so we're going to talk about fermentation and anaerobic respiration. Alright, so what we've been discussing is the aerobic cellular respiration pathway. Alright, now notice that in terms of the major committal step, for pyruvate to enter the mitochondria and go through the process of forming acetyl-CoA and that entering the citric acid cycle, you need there to be oxygen present. Alright, so if oxygen is not present, alright, an organism is going to switch to either anaerobic respiration or fermentation. Now, different organisms are restricted based on the particular genes, proteins, and enzymes they have for certain metabolic pathways. Alright, so some organisms are strictly restricted to just aerobic metabolism. Others are restricted to fermentation. Others can fluctuate between using aerobic metabolism, fermentation, or anaerobic respiration. Alright, so when you're talking about processes that occur in the absence of oxygen, alright, this pyruvate molecule can be shunted into different locations within a cell. Alright, so looking at anaerobic respiration. Alright, so usually, if you recall from aerobic respiration, oxygen functions as the final electron acceptor, alright, of cellular respiration. But in this case, if oxygen is not present, alright, some organisms have other chemicals, molecules that can function as the final electron acceptor. Now, a lot of times these molecules contain oxygen. Alright, so things like nitrate, for instance, or something like sulfate. Alright, be two instances. In this case, the nitrate or the sulfate would act as the final electron acceptor. Alright, so here you have nitrate plus NADH. Here, nitrate reductase is going to catalyze the removal of an oxygen atom from nitrate. You're left with nitrite and water and your now oxidized electron acceptor molecule, NAD+. Alright, so here, looking at the conversion of nitrate to nitrite. This enzyme nitrate reductase allows the pumping of protons, alright, across the membrane of the cell. In this case, it could be the mitochondria. Alright, and now these protons flow back through the ATP synthase enzyme and you generate ATP. All right. Here you have sulfate plus some organic molecule. You have a sulfate reducing organism, like a bacterium, that can take the sulfate and it's going to be converted to sulfide and CO2. Alright, so this is very similar to what's happening with nitrate reduction. Here you have sulfate acting as the final electron acceptor in place of nitrate. Alright, so here you have NADH dehydrogenase, alright, so which is an enzyme that accepts your little electrons from the reduced form of NADH. This allows for the pumping of protons into this external space. The electrons are shuttled between various membrane proteins, and here you have your nitrate reductase enzyme, which allows for the transfer of the electrons to nitrate, generating nitrite, which allows for now you have this buildup of protons. Those protons flow back through the ATP synthase protein, and you make more ATP. Alright, so in this case, the nitrate functions as the final electron acceptor in place of oxygen. Alright, now looking at fermentation. Fermentation is a process that's used by cells to recycle NADH back into NAD+. Alright, because here, as you convert, for instance, something like an organic compound like glucose into other intermediates, in this case something like G3P, and you're forming your oxidized compound of pyruvate. Alright, so basically you're looking at this process of glycolysis. What you wind up doing is you wind up transferring the electrons from this organic compound to NAD+, forming NADH. Now, if you're talking about a cell that has a mitochondria, or even a bacterial cell, the electron transport chain in the absence of oxygen is going to be temporarily turned off. Okay, so there's only a finite amount of these NAD molecules in a cell. Alright, so once they're all reduced to NADH, there are no more. Right, and the cell's only means of generating ATP from the central carbon metabolism pathway involves the NAD+ molecule becoming reduced to NADH, so then you can also make ATP through glycolysis. Alright, so you have to recycle the NADH you do have to make more NAD+.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are two types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Alright, so looking at fermentation. Alright, you have alcohol fermentation and you have lactic acid fermentation. Alright, we're going to look at each of these two processes by themselves. Alright, alcohol fermentation is going to occur in yeast. Lactic acid fermentation are going to occur in our, for instance, muscle cells. Alright, so looking at alcohol fermentation. Here we have our pyruvic acid, alright, which is the byproduct of glycolysis. Now this pyruvic acid, alright, notice here you have your little carboxyl group. Alright, so again, carboxylic acid functional groups make the molecule acidic. Alright, so that's why we call it pyruvic acid. Alright, so you have a decarboxylation reaction. Alright, where you cleave off this carboxyl group, you're left with CO2 coming off, and you're left with acetaldehyde. Now, this acetaldehyde molecule is then going to be reduced to alcohol by NADH. NADH gives up its electrons and hydrogen atoms to acetaldehyde. The NAD molecule becomes reduced to NAD+, and you're left with alcohol as a byproduct. Alright, so the whole purpose for which yeast use fermentation is so that they can recycle this NAD+, which can come back into glycolysis so that the yeast can make more ATP through glycolysis. The alcohol is actually merely a byproduct of this process and can be potentially lethal or toxic to the yeast cells. Alright, so here you're looking at lactic acid fermentation. Alright, so in lactic acid fermentation in muscle cells, the pyruvic acid is going to be reduced to lactic acid. So NADH is transferring electrons and hydrogen atoms to pyruvic acid, and you're left with the oxidized form NAD+, and the reduced pyruvic acid as lactic acid. Again, it's an acid because you have this little carboxyl group, which is acidic, which makes this molecule an acid. Alright, so looking at muscle cells. In the absence of oxygen, when you're furiously exercising, your muscles go into a period of oxygen debt when you can no longer provide your muscles with sufficient supply of oxygen to carry out aerobic cellular respiration. Your muscle cells switch to lactic acid fermentation in order to generate energy. Alright, so here your pyruvic is converted to lactate or lactic acid. This lactate will then enter the bloodstream, is transported back to the liver, where it can be reconverted back into glucose, which can then be delivered back to the muscle cell. Now, a lot of the soreness associated with vigorous exercise, the burning sensation, is thought to be due to this buildup of lactic acid. Alright, so in terms of fermentation and its various applications, alright, we utilize different kinds of organisms and their ability to ferment different molecules, like sugars, and their byproducts. Alright, so we have the use of certain fungi and bacteria in making cheese. Alright, for instance, you have the production of wine and beer through alcohol fermentation. You can generate things like acetic acid or vinegar, ethanol, propanol, acetic acid through various fermentation pathways, as well as some organisms as a result of fermentation. You actually have the development of gangrene, gas gangrene, due to the toxic byproducts of certain microbial organisms that are strict anaerobes. Alright, so looking at the entire process, alright, going from respiration, using cellular respiration and converting glucose through glycolysis to pyruvate. Pyruvate is converted to acetyl-CoA. Acetyl-CoA enters the Krebs cycle, and all of these reduced electron carriers are then shunted to the electron transport chain in the membrane, the mitochondria. And now, as these membrane proteins in the intermembrane mitochondria transfer, you have to transfer these electrons from one protein molecule to the next. So you have a series of redox reactions, which allows for the pumping of protons across the membrane. You build up proton concentration. Those protons flow back through this ATP synthase molecule, protein, to allow for the production of ATP. Alright, and when you're talking about aerobic cellular respiration, final electron acceptor is oxygen. For anaerobic respiration, it could be nitrate, sulfate, carbon dioxide, and then talking about fermentation, you have pyruvic acid or pyruvate as a final electron acceptor, or some other organic molecule, like, for instance, acetaldehyde. Now, we've only talked about this process here. We've talked about the breakdown of sugars and their entry into cell respiration. What you have to also keep in mind is that your fats, your lipids, your proteins can also be broken down, metabolized, and used and shunted into central carbon metabolism. Alright, in various stages. Alright, so keep in mind that you have, for instance, glyceraldehyde 3-phosphate. Well, this glycerol can come in and make up this product here. Your fats can be used to make the acetyl groups. Your amino acids can enter the production of acetyl-CoA, or they can enter citric acid cycle in different stages. And that's when your body needs to break down these foods for energy. Alright, now, what happens when you have an excess of these things, or you have a need to assemble larger biological components? Well, you can take sugars and form glycogen or starch. You can form nucleic acids. You can generate phospholipids and fats, all right, by shunting different intermediates from a central carbon metabolism pathway. All right, you can siphon off amino acids that are used for the assembly of amino acids into proteins. All right, so in this case, you have a pathway that's both catabolic and anabolic. All right, so that's why we call it amphibolic pathway. All right, because you can break down molecules as you need to produce energy, but then part of that process is involved in taking the individual components, building blocks, and making larger biological structures like proteins and carbs that are used to make up parts of a cell.